Okay, good morning, everybody. I'm so pleased we have a good turnout. This is our first candidate for our new botanist position at EKU. Dr. Abbott, Richard Abbott, earned his undergraduate degree from Berea College, just down the road, and his master's and PhD from the University of Florida. He's currently an assistant professor at the Department of Biological Sciences at the University of Arkansas in Monticello. Dr. Abbott has broad skills in plant identification and has worked throughout the southeastern United States, as well as the neotropics as a postdoc, botanical research contractor, consultant, visiting lecturer, and horticulturalist, and has authored and or co-authored over 30 publications. Other previous employers include the Missouri Botanical Gardens, the New York Botanical Garden, and Murray State University. Please join me in welcoming Dr. Richard Abbott.
Hello, thank you for being here. So as you know, you're here for me to give a research seminar, and I've kind of started with this somewhat silly title of go ahead and remove this thing out of the way here.
You just ignore, or I mean,
Well, it's,
you know something better, but
there's a way of making, you can hide the floating meeting controls, so we'll get those out of the way. Awesome.
Can you get the recording on?
And so why I started with this kind of silly like Argh, there's a milkwort on my P is because I want to actually kind of not just do some hardcore presentation of my research, in which case the title would have been, Phylogeny and Biogeography, North American polygalaceae. But what I want to spend the first few minutes doing here, oops, that does not want to advance. So,
again.
there we go. Is I actually want to spend a little bit of time kind of talking about integrating research and education, and particularly in my experience, when I talk about kind of education, I'm also not just meaning in the classroom in academia, but actually kind of community outreach. I'm talking with the public, because if we don't share our knowledge, if we don't have the support of the public, then what we're doing is just not being very useful, right? And so to be meaningful and most practicality, we need the public to understand what we do and why we do it. So I'm a firm believer in wherever I'm teaching, whether it's Kentucky, Illinois, Arkansas, Florida, anywhere, is teaching locally, but with a global focus, and trying to understand global patterns and global significance behind what I do here. And I'm well aware that most of the time I'm not actually a training botanist. It's great to work with other botanists, but most of the time what I'm doing is actually just training general students to have an understanding of plants. That's the primary thing that I'm after, is just trying to get students to understand plants. And have just kind of an educated populace understanding what plants do for us. And most people I talk to just couldn't care less about plants. So it's just not their interest, but they often like animals, right? And so I asked them, I said, well, what are the lions and tigers and the frogs and the golden tanagers and things like this that you like? What do they need? Now we see this golden tanager here is actually feeding on some Witheringea solanaceae, a tropical tomato relative, right? And these maps that we see here showing the vegetation, different colors correspond to different types of vegetation. Ultimately, what it boils down to is if we're going to take care of these other organisms, if we're going to take care of the planet, it's about managing the plants. Right? This is what wildlife management is about. It's largely about vegetation management. There's more to it than that. But ultimately, we need an educated public that understands why plants matter. So again, a huge part of what I do is try to get the people to understand that plants are providing oxygen, food, the energy. It's amazing that plants can convert sunlight and energy into chemical energy in the food. And the food isn't just about the energy, it's also about the fixed carbon molecules, right? Organic life made of organic molecules. Well, plants are doing this, right? Some people's eyes still glaze over at that, even when I try to explain this. And I say, well, y'all at least can care about poison ivy, right? So if you think you might be allergic to poison ivy, if you might want to be cautious, you might want to learn to recognize poison ivy as opposed to something like Virginia creeper. And so the public likes learning about plants, I find, sometimes more than the students in the class like learning about plants. But my approach, I came across this a few years ago, this idea of the zone of proximal development here. Every student is different in my eyes. I know that we have a one-size-fits-all in terms of attendance policies and grading and such, but each student comes to the class with their own skills. And every person I interact with has their own existing knowledge. And I like trying to expand what they already have here and try to realize that what I'm trying to accomplish is not a one-size-fits-all kind of thing, but it's going to be different for every student I interact with. And one of the things that I always kind of find is a great convenient starting point is something as simple as a tomato being a vegetable. Well, there's reasons why we call it that, but it's got seeds, therefore it must be a fruit, right? Stuff that we call nuts aren't actually even really nuts, right? And so this is kind of a great starting point because most people, what little bit they might know about plants actually turns out to frequently be wrong. And it gives me something to work with, right? I like being wrong, actually. I like making mistakes because why? I get to learn from those mistakes. Making mistakes is a growth opportunity, it's a learning opportunity. And so there's so much vocabulary involved to talk about plants. If I'm going to talk about plants, I have to use words like blade, petiole, compound, and students need to know what I mean like this. And so I stress to my students, botany is a language. Botany is a language. And it doesn't matter. I've got in Spanish here. I just gave a 3-day class. It was a 6-hour course in Spanish online for a Congress in Colombia for some friends there. And it doesn't matter if I'm speaking Spanish, Portuguese, English, French, any other language that's out there, right? Bottom line is botany is very comparable. It needs to be studied like a language. You have to learn the basic vocabulary to talk about plants. And once you've done this, once you've embraced this and you accept it, okay, I'm going to learn the language of botany here. There are patterns that start to emerge. And so the Asteraceae, the largest family of flowering plants, 30,000 species, a tenth of all flowering plants are this family, has a motif, a pattern of having an involucrate head. Well, head is an inflorescence type where the flowers are tightly packed on an intercessional and expanded receptacle, right? Talking about Asteraceae can be reduced to 2 words, involucrate head. Is it more complicated? Oh yeah, in a systematic class, there's a lot more detail, right? But for the general public, if I can get them to see these dandelions that they've grown up with and known their entire life as having an involucrate head, and that's a pattern, then I can very quickly have them start seeing other patterns. And the beauty of this, you look at the map down here below where the reds and purples and blues represent the most biodiverse places on the planet. It turns out that even in these hyperdiverse areas, a dozen or so families can make up more than half the species. And we're talking specifically in this case for this study, we're talking about tropical, lowland, woody plants here. If you go to the most hyperdiverse places on the planet, the tropical, lowland, woody plants, are more than half of them are going to be a dozen families, right? And those same dozen families can make up more than 2/3 of the individuals. Learning these common patterns, learning these motifs has become kind of one of my goals in teaching, is to help expand students' minds to understand the patterns that are out there, right? And most students get aggravated. It's just too many words, too much going on here. At the same time, how else are we going to combat plant blindness, right? This idea that most people don't see plants, don't understand plants. It's just a sea of green, as one of my students said. He said, you don't seem to understand how, when I look at this, I don't see a plant, I just see green. I don't see petioles and I don't see flowers, I just see green. And my point is by learning the words, you can actually start to articulate, you can start to break the green apart, right? And so the idea behind this kind of silly title of Argh, there's a milkwort IP, was embracing the fact that you don't go to the general public and start talking about phylogeny and biogeography. Those aren't words that you would connect to the public with, right? But I can say these plants are really cool. I can say plants are awesome. You know? And so, and these kinds of silly titles with these Toy Story aliens and stuff. I did a lot of kind of community outreach when I lived in St. Louis for 6 years. I worked with a lot of STEM groups through the St. Louis Academy of Science. And I loved reaching these younger kids. Why? Because by the time most students have come to college, they've already decided what they want to be, what they want to do. And very few are actually interested in plants. So I like stepping back and getting them when they're even younger and saying, aha, here's this cool hook so we can talk about. And this is where the language and the travels come into this all of a sudden. So this is in Portuguese here, but the Botany are the vegetation types in the lowland Amazon. And you just ask a group of kids, do fish make nests in trees? And of course, they're going to say, well, no, fish don't make nests in trees. And then you explain how this area floods 5, 10, 15 meters of water every year for several months, and fish come along and make nests in trees. And some of the kids, all of a sudden, you just hook them. They're like, oh, cool, that's really cool. Fish making nests in trees, right? And so this is what I love about teaching at that level. And part of what I've realized is that even in college, there's some room for a little bit of silliness, a little bit of connecting to the students in a way that draws them in. And so when I was working through the Academy of Science in St. Louis, and I was doing a lot of the inner city kind of STEM stuff, they said, we want you to promote education. These 10-year-old kids and 12-year-old kids, they know about poverty and unemployment. They're familiar with these. We don't have long discussions about these kinds of things here. But I point out to them that if they want to make their lives better, they need to be educated, because as voters, we have some control. What little bit of power we have in our society is based on our ability to vote. I also point out that their lifetime earning potential is going to be tied to their education skills, right? And so education is super important, right? And so this is something we can't forget, that part of what we're doing with research is actually building our knowledge base, and we need to share that with the public. We need to help the public be educated about what we do. And so again, working with kids groups, it's so much fun to take a simple example like cats and dogs. And if I ask this question in this room, I suspect that I would have the highest percentage of people who know good answers here. How do you tell cats and dogs apart? And it's a fun exercise to do that with younger children. But I'll ask college age kids this, and it's very rare. Not more than 1 in 100 kids points out something like retractable claws, right? They're talking about what cats eat fish and dogs eat, and it's just, and it's not that the stuff they're saying is wrong. It's not that the stuff they're saying is bad. It's just understanding the deeper level. There's a difference between what you imprinted as a kid. No one ever taught you cat versus dog. They just pointed and said, that's a cat, that's a dog. You imprinted that knowledge, right? Coming to college is about learning to diagnose, learning to critically assess what you know. And that's where the research fits in, right? This pursuit of knowledge, this research to learn needs to be integrated with the education. This keeps freezing up a little bit, so let's get that going. And so back to the title here, back to what we're talking about here, this idea of milkworts and peas. Well, let's just go and clarify that milkwort is a common name for the polygalaceae. It's a family of plants. Wort is just Old English for a plant. It's just, again, Old English for a plant. And milk, there was a belief long centuries ago, people thought that the cattle that ate milkworts were going to increase their lactation. It's a mistake, it's an error, right? So it's not true. But here we are hundreds of years later, stuck with a common name that means literally nothing, because it was just an error that was made in the past. But the interesting thing here, and why it's kind of fun, and this is starting in on the actual the science part of what I've got here, is the flowers of milkworts and peas. The flowers of these two families, peas are part of the legume family. The papillonoid legumes have flower parts called wings and keels and banners, and so do polygalaceae, right? Polygalaceae have very similar flowers. Historically, it was seen as convergence. People said, oh, it's just a completely unrelated lineage, it's a conversion on the same flower. There was some sort of natural selection pressures driving for this, right? We'll come back to phylogeny and biogeography in a second here. But it turns out that modern phylogenetic analysis, and phylogeny refers to the study of relationships, right? Modern studies of relationships showed milkworts and legumes were actually related. When I first started working with this family, we didn't know this. It wasn't known that this was the case, right? And so this is a shock to a lot of people. So it turns out, instead of being convergent and completely unrelated lineages, it's actually more like a parallelism of related lineages developing similar flowers. The homology of the flower structures are different. The wings of legumes are actually petals. The wings of milkworts are actually sepals, right? Homology is different, but similar environmental pressures driving for similar flowers, right? So it turns out that the polygalaceae are related to legumes. And that's kind of a weird, novel thing that's only been known for 20 or 30 years now. In the old days, what were the milkworts related to? A lot of families that the DNA says, nope, not related at all. We don't need to worry about all these names here. I'm not going to be a test on or whatever. Just wanted to make out that I wanted to introduce this idea of artificial groups. It turns out that about 30% of what we thought we knew with respect to families and orders was wrong. It was based on artificial groups, artificial misunderstandings. And so imagine for a second now that you decided to go ahead and just put all the winged animals together into a group. Is that useful? Maybe, maybe not. Is it natural? Definitely not natural, right? But if you were some alien life form that came to this planet, you were just trying to catalog the organisms and put names on them. If you were just a novice who's starting out, would it be useful to teach people about winged animals? I think so. If the goal is to put names on things, right? Because we all know that bats, mammals, the birds, and the last dinosaurs, reptiles, and then we have these inverts here, right? So we know that they're different. We know it's not natural. We don't want to classify organisms based on natural morphological similarity here. But as for practical ID purposes, natural groups are often quite useful, quite memorable, but sometimes natural groups can't be diagnosed using morphology. Sometimes we want to fall back on artificial motifs to put names on plants. So I'm not going to talk about this in detail. There's a lot of stuff I'm skimming over here, though, that if it were a class, I would go into much more detail. I wouldn't cover as much, try to cover as much. And if it were the general public, I might not go into quite exactly the same detail either. So I'm kind of hybridizing the two here. But this idea of a clade, understand, we've had a paradigm shift where we're actually now using cladograms to understand relationships here. And what cladograms boil down to is understanding sister group relationships. So if we look at this example here, what this is telling us is that 4 is sister to 5. And this group 4, 5 is sister to 3. And so cladograms tell us about sister group relationships. They tell us about patterns of relationship here. And when we talk about natural groups, we're talking about a lineage or a clade. We're talking about a monophyletic group, a group that contains a common ancestor in all of the descendants. So if you all of a sudden took these organisms here with the marginal cilia, the marginal hairs here, these projections on the side, and you thought that they were related because of those projections, there's nothing inherently wrong about that. But if this is the true phylogeny, if this is the true pattern of evolutionary history, lumping those two together into a family would create an artificial group, right? And this is the point. About 30% of our plant families and orders turn out to be artificial. And this is why so many names have been changing in recent years. And this is what was kind of driving my own research questions, was to trying to figure, okay, now that we're linking classification and phylogeny, now that we're trying to talk about having monophyletic groups, now this whole paradigm shift that I just talked about exists here. We're going to focus on monophyly and synapomorphies, these shared derived features, and focus on single lineages and such. And we're going to have a transparent analysis of all the data, not just someone's personal opinion here. Now that we're using cladograms the way that we do here, I want to know what are the subgroups and milkworts. And one of the questions that came along here is asking yourself this other question: are species biologically real, or are they just a human construct? And this is something you could debate for the rest of your life and probably not settle on an answer for. But underlying this is there are some philosophical, theoretical issues that would need to be resolved. But ultimately, for me, I just want to know what are the subgroups of milkworts? What are they basically? And so this ties back into the phylogeny and biogeography part here. My original goals here then were to say, okay, what are some modern hypotheses of relationships within the subgroups of the milkworts using various lines of data? I wanted to take one of those groups and revise it, monograph it, study it carefully. And it was just, it wasn't sheer coincidence that I chose a group that happened to be endemic to the Caribbean. So I got to island hop in the Caribbean for a few years. That was fun. And I wanted that to culminate in a treatment of the flora North America, of the milkwort family. And what I quickly realized is in order to understand North American polygalaceae, I actually needed to understand the global context. Well, I couldn't just do North American isolation. I had to go ahead and step outside North America to also understand the bigger picture. And so my project all along was kind of just a bigger picture than global, turn into bigger picture global look at systematics. And specifically, we were looking at the biogeography, disintegration of polygala, trying to figure out what was going on here. It turns out that polygala was a bit of a mess, right? It was an artificial group here. And so when I first started, we had just found out that we had just gotten the first phylogenetic analysis saying that the polygala was not a natural group. Right? All these genera here that have P after them were polygala when I first started my work, right? These were things, look at all the things that polygala was, right? And this was awesome. I was like, yay, somebody else is helping me out. Somebody else is doing the same thing, right? It's nice to have a collaborative world where lots of people are working on things. So I was excited to see that somebody else was saying, hey, yes, polygala is not monophyletic. Awesome, great start. We know that polygala is artificial. It turned out when I started looking at the sections of polygala in North America, again, with this North American focus, and I will point out, I actually started all this later in life before I even started a PhD. I wanted to pursue this stuff of curiosity. I actually went back to get a PhD because I didn't know how to do this research without having some more training and education. But this is a project that I started a long time ago, long before I even started a PhD. But I was trying to figure out what's going on with the North American species here. There are lots of different groups. Turns out that within this genus polygala, more than 1,000 species traditionally, there are actually more than 50 different named groups. And it was really confusing trying to sort out, well, what is what and who's related to what. But I went out and I spent several years actually traveling again. Before I even went back to graduate school, I spent several years traveling across North America, collected 47 of the 53 species in North America, got DNA of them, ran this analysis, and I was like, yeah, I've got this analysis of North American polygalas. And then I was, but something's missing, right? The global context was missing. Right? You can't sort out relationships in isolation. You can't just draw some artificial geographic boundary and call that a phylogeny. Right? You have to look at the big picture patterns here. One quick aside on this for anyone who may not be familiar with the use of DNA data. Your DNA, as you're aware of a nucleotide, it's got these 4 basic nucleotides, right? ACs, Gs, adenine, cytosine, those kinds of things, right? You can actually sequence the data, and it's like looking, saying, are the flowers red, or the flowers white, or the leaves opposite, or the leaves simple? And so they're characters, right? You've got these basic characters here. You can line up the data, you can align the sequence, and essentially, the more similar the species are, the more their DNA is going to be the same, the more closely related they are, basically. And so this is not meant to be the hardcore details here. Don't even try to make sense out of most of my slides. There's too much information on them, and this is no exception. What I wanted to say is I expanded from having the 50 samples from North America to having more than 500 samples globally, and said, okay, now that I've got these 500 samples from all over the world, and I've got this genetic data here, what are the patterns that emerge? And I did the stuff that scientists are supposed to do. I manipulated the variables. I said, okay, what if, chloroplast data, so the chloroplast, the photosynthetic organelle in a plant cell has different DNA than the nucleus, right? And so I said, okay, do they tell the same story? If you've got different DNA in a chloroplast than you do in the nucleus, do they tell the exact same story or not? And then I also experimented with the number of taxa. What happens when you have 50, 100, 200 taxa? And essentially ended up settling on, okay, here are the clays that seem to be consistently supported. And so I was finally able to say, aha, I've got my own my own evidence telling me which genera I should be recognizing, right? So what I want to do now is stop and kind of just go back and actually pat. I've been talking about the milkwort family. What I want to do now is I've kind of set the stage for the preliminary work that I did. And now I want to go back through and quickly run through some of the family itself, right? And I'm not going to read most of these words to you. This is my understanding. This is being recorded. And anytime I know something is available for re-review, I always shove in as much as possible for people to use. But when I started 10 years ago on this, we were recognizing 19 genera of polygalaceae. We're currently recognizing 29. We actually just submitted a manuscript, but there's going to be a 30th genus. We're still working on sorting out the genera of polygalaceae here. What do they have in common? What do we actually find here? Though we'll talk more about that in a second here. But it turned out that polygala vary from being annual herbs all the way to canopy emergent trees and everything in between, right? This is just the genus polygala did this. So we've known for a long time that this family that this genus was artificial. I chose it in part because it's worldwide, and guess what I wanted to do when I was younger? I wanted to travel, right? I wanted to go see the world. I wanted to get out there and move around. As a botanist, it's been awesome traveling the world, studying this group. Right? As a family, to characterize them, I talk about 5 basic ID motifs, and 5 basic vegetative patterns are the habit. Most polygalaceae in North America are herbs. There are shrubs out west. I talk about leaf composition. These leaves are simple. The blade is not divided into segments. I talk about leaf arrangement. It's a 1 leaf per node, 2 or more. Most polygalaceae have alternate leaves. There are exceptions. And then I talk about the margin. In this case here, the margins are entire, meaning they have no teeth. And then as far as the stipules are paired outgrowths at the base of the blade, base of the leaf. Polygalaceae don't have stipules, except turns out there are a few polygalaceae that actually do have opposite world leaves. A few, not many, but a few do that. And several different lineages of polygalaceae actually have these kind of glands in a stipular position. And now that we know that milkworts are actually sister to the legumes, and legumes are characterized by having stipules, chances are that the polygalaceae ancestral either had stipules or at least have the genetic preadaptations to develop stipules, because half a dozen different lineages of polygalaceae actually put out stipular glands, and a couple even have kind of stalks, obvious looking stipules. But generally speaking, the family has always been described as not having stipules, except for the ones that do, basically. The flowers we've already kind of talked about, these distinctive, bilateral zygomorphic flowers here with the distinctive wing. So these are the wing petals. Here's the filament sheath, the male part of the flower. Here's the keel petal with the crest. Very, very showy flowers in a lot of species. But the floral variation is quite diverse. A lot of diversity in the flowers. And this is still just polygala in a strict sense here. Tremendous diversity in the flowers, right? The fruits of the family, particularly the subfamily and tribe that involves polygala, they're almost all capsules, which is a dry fruit that splits open to release the seeds with 2 or more carpels. And they tend to have a single seed per, or they always, in this tribe, always have a single seed per lot in this group. The seeds tend to have a caruncle or lysome, which is a fancy way of saying an arrow, which is basically saying that there's a lipid-rich food bud. The plant basically rewards ants and such for coming and taking the seeds away and dispersing them, and then feeds them with this fatty tissue. There are a few exceptions. I want to make it clear that in the time that I have here, I'm not trying to cover the entire diversity, right? Here's an example of a pan-tropical genus called Secredaca. They're lianas, and they actually have samaras for fruits, right? 1-seeded winged fruits, right? So there are exceptions to these features I'm talking about, but we're just going to gloss over those details for now. Polycoporate pollen characterizes the family that has multiple pores, multiple grooves. And it turns out that now that we have a better understanding of the genera, we're actually able to go back in and map and say, hey, wait a minute, there's actually different types of pollen based on the clades that we're now finding based on DNA and morphology. So pollen morphology it turns out to be one of the features that was historically used to identify the polygalaceae. Those other families that were artificially lumped into with polygalaceae, it's because they also had polycoporate pollen in most cases. So back to the final part here, the North American species here. What I want to do now is run through a handful of the North American species and talk about the biogeography side of this. The one in green here, Monina, has never been a polygala. This has been its own genus, but the other 5 that we have here are historically polygalas. In fact, this polygala in a strict sense is being split up. The old world polygala have the type species, the New World polygalas are in a different clade now. So unfortunately, I've fought this for 10 years, but the bottom line is I have to accept the data. I spent 10 years double, triple checking, and it's just overwhelming evidence that the New World polygala are not closely related to the Old World polygala. So the New World polygalas are going to become the genus Synaga, which again I'm not happy about, but the evidence makes it clear, right? So let's run through these quickly here. As far as biogeography, again, this could be an entire semester long course talking about this. Most people are aware that continents move around, although in the old days, people didn't believe that. Right? You go back 70, 80 years ago, textbooks kind of made fun of continental movement, right? Continents don't move. But we know that they do. And a lot of people wanted to jump on this idea of vicarious biogeography, that the idea that the plants moved around and rafted and found their current distributions by moving on continents. Most of the continental movement took place so long ago, though. Studies have shown continental movement has almost nothing to do with most modern plant species distributions. Right? It's a historic thing. It affects some ancient lineages, but it does not have anything to do with most modern species distributions. Weird thing, though, is you keep moving forward, and it turns out there was tied to sea level rise and glaciation cycles and stuff. There was actually a seaway in the middle of the US. And so again, I'm just laying out some of the factors that you might want to consider when you're thinking about biogeography of species in North America, the fact that we actually had this intercontinental seaway. Something like the end of the Cretaceous Tertiary, the KT boundary, like 65, 66 million years ago, this giant bolide impact and created the Chicxulub crater right off of the Yucatan Peninsula, right? Would have wiped out all life in that area, right? There would have been some tremendous devastation. So we can think about all that, but all this stuff is so far in the past, it has almost no impact on modern species, right? Most modern species have just in the last 10, 20, 40 million years, and all this kind of stuff, which is great, people talk about it all the time, probably has nothing to do with current patterns. Sea level rise, and here we're looking close up at Florida here, and the fact that if you go back 10, 12,000 years ago when the first Paleo-Indians supposedly came to North America, the Gulf Coast of Florida was 100 miles further west, right? And when sea levels rise by 50 or 100 feet, Florida gets a lot smaller. When sea levels drop, Florida gets a lot bigger. And so the idea that what we're planting here is a seed of an idea that biogeography could be studied mechanistically, but what we're doing right now is still building the phylogenetic framework for even knowing which groups to talk about. So we don't have any answers with respect to biogeography other than saying that we know that we have at least a dozen different a dozen different lineages of polygalaceae in North America. This is the Monina. This is the one we talked about here that was never a polygala. This grows in these kind of oak pine savannas out in the western part of the US. Our Monina is part of this kind of subgroup that has an amphitropical disjunct. So our exact species that we have that barely makes it into the arid Southwest stretches through Mexico, and then it's found down here on the south side. So amphitropical means on both sides of the tropics, right? Both sides of the equator. This is a well-documented pattern. There have been lots of recent studies looking at other genera, other groups to this. And the mechanisms behind them have been different in every case. It wasn't a single association that moved. There have been different answers in every single case that's been studied. But anyway, so this is kind of unusual biogeography. Hebecarpa bodiera, when I first started this and people first started splitting up the genera, they lumped these two together. And I said, I don't think they ought to be lumped. They didn't seem like they should be lumped to me. I said, these look different to me. And so I actually chose bodiera as the group I was going to study. One, because it was a group of Caribbean shrubs and I wanted to go to the Caribbean. But also primarily, I wanted to figure out why were these being lumped? They look different to me. I didn't think they should be lumped. I didn't think they should be the same thing. But if you pick up the Florida of Ecuador treatment, their Hebecarpa is being called bodiera. Shouldn't have been, in my opinion, but that's the way it was. So that motivated me to step up and say, okay, let's study this. It turns out that Hebecarpa, and I'm grossly simplifying and leaving out a lot of details because of time. This group called Hebecarpa is characterized by having a deciduous perianth. You look at the fruit, these capsules, and the perianth is the name for the two outer whorls of the flower, right? The sepals and the petals. The perianth is completely deciduous in this group called Hebecarpa. We move on to bodiera, and it also had the deciduous perianth. Okay, it's got that going. But these are vergate shrubs. Vergate means like wand-like. And so instead of being kind of small, suffrutescent herbs in the desert, they're actually tall. They can be over a meter tall, sometimes 10, 15 meters tall. The lateral wings, the wings on the flowers are actually very reduced. And then they actually have capsules that open up with bird dispersed seeds. So there's some interesting stuff. There's also some anatomical differences and DNA differences. But the bottom line is I chose bodiera to monograph, to revise, because I wanted to go down and study some island biogeography, basically. And I ended up settling on 9 species. And this is a whole other talk. I'm just going to gloss over here. And the reason I'm talking about this in detail at all was just to stress, in terms of my types of research, what I like to do. Field work, floristics, the DNA work, the phylogenetic analysis, but I also in the past have used plant anatomy as an important tool. So I studied the anatomy of the leaves. There was this fascinating case here where there were these two entities being lumped together. This is an endemic to northeastern Cuba, Bodiera alternifolia. Notice how in this case here the hairs, these species were lumped together because they had dots in the leaves. See these dots in the leaves here? If you look at these dots, they're actually pits in the cuticle on the upper surface of the leaf. The species they were being lumped with had dots in the leaves as well, but those were actually raised pustular bases. They were different dots. They didn't have the same homology. And so they shouldn't have been lumped together. In fact, turns out DNA says the same thing, and I found a bunch of other characters to support these as different species. And so this idea of exploring cryptic species is part of what I like to do research-wise, is trying to sort out what are the species, what are the genera. It's part of what motivates me. And sometimes plant anatomy is a useful character. Here we're looking at its stem anatomy. I studied the detail, the floral details. Here we're looking at the perianth and the andrecium, the male part. Here's close-ups of the gynecia. And so again, spent years studying the micromorphology. I even went so far as doing SEM work. Spent several weeks in the SEM lab studying various features here. So I've just mentioned that to point out that part of what I use in my tools is just dissecting microscopy, compound microscopy, and also scanning electron microscopy are all tools that I'm familiar with. Studied the seeds as well. And what I ended up concluding was 9 species. What was really dissatisfying, though, is that the 6 of the species were supported as clado species, as monophyletic lineages, and 3 of the species came back as completely interdigitated, right? And what does this mean? This is that question I asked earlier about, are species biologically real? If so, if species are biologically real, then species don't have to be monophyletic. Because if you think about how species originate, species normally do not originate in a dichotomous monophyletic way, right? But if species are human constructs, which most other ranks are, we now have to go ahead and say, okay, this is unacceptable. This is untenable. And what's really needed is more study, right? More study trying to figure out what's to be done in this group here. And this is something I hope to follow up on. But ultimately, this is where I kind of ended. And then I was basically told, okay, time to graduate. You spent 7 years on this, time to graduate. So, and that's where that story got left. But I want to follow up on that some more. Finishing up with the North American stuff, we have a Rhinotropus group that's got 12 species in North America. And this is a cool story because it starts in the redwoods. The base, almost early diverged species in this clade are actually growing mesic woods in the western US, but then they diversified into the deserts. So they're in the redwoods. There's a couple species like Rhinotropus californica that you can find. But when they moved out into the deserts, they turned into these thorny shrubs, basically. And what characterizes Rhinotropus, if you think about a rhinoceros, rhinoplasty, nose, there's a beaked kill. So the kill petal actually has a beak on it. And that's what characterizes this genus Rhinotropus. These species that we have mostly kind of suffrutescent, low shrubs growing in the areas out west. Asamea turned out to be a clade that we only have one of in North America, but it's widespread from in Latin America. And Asamea, what defined this genus, turned out to be that the lower 2 sepals are fused together. So this is a bilobe structure here. This is 2 lobes here. This is actually 2 sepals fused together. And so notice that every single genus had different characters. Historically, people just would choose a character and say, this is the character I'm going to use. Now what we do is we actually look at what the phylogeny has to tell us and try to then use that to reciprocally illuminate the DNA morphology, provide reciprocal illumination. And so notice how every single genus has different characters that define it. And that's part of the modern paradigm, which I love, is that we don't have to be constrained by one person's thoughts, right? What do the data have to say? Polygaloides is one of our prettiest ones. We have 1 species here, and this is a bizarre clade because its relatives are in northern Africa and Europe. It's also a mess because it turns out that there's 2 other lineages. This might actually turn out to be 1 giant genus, or it might be 3 genera, and we're still working on that. That's one of the projects that we haven't resolved yet. It depends on which analysis we look at. But gay wings, as this thing is called here, this is known from one county in Kentucky, Laurel County. In fact, it was an EKU grad student who found this. And this guy, Randy Mears, years ago, actually brought me this plant. He's like, what did I find? I was like, I had no idea. I've never seen that in my life. And I used the Florida, West Virginia to figure it out. And so this dot right here on the map was actually a spot I can take you right back to that I discovered with an EKU graduate student, some time back. And so this group is hard to characterize. Compared to North American stuff, has the largest flowers. It's got 1 to 4 flowers in it per raceme. The leaves are clustered at the tip. It's got a different growth form from any other North American polygala, but its relatives are actually in Asia, Africa, Europe. It needs to be studied more carefully. And so this brings us to the last group here, the polygalas, which historically we were looking at 31 species in North America. We're still working on this. In fact, we have a manuscript in prep right now. I'm sorry, in press, it's been submitted. So I should say it's in review, I should say. Hopefully it's accepted. But polygala itself is still super diverse and hyper diverse, and we're still working on it. So again, you can't read the details. I always found scientific talks where people try to just read their cladogram to me to be really boring. Wait for the publication or come talk to me, and I can show you better details. But what this boils down to is that in North America, we have one introduced, the type species of the genus Polygala vulgaris looks very much like this species here, has been introduced in a few spots, but otherwise the rest of our North American polygalas are not related anymore and are going to become a new genus. And so we have at least 6 different lineages within Polygala. This species here, Polygala hemipterocarpa, and again, I like etymology, I like words that make sense. Hemipterocarpa, hemipterocarpa means half-winged fruit. Here's the fruit. This is what characterizes this clade. This clade is sister to the rest of the New World species. And it's got the fruit that has a wing on one half of it, right? So again, I like names that make sense here. So this is a representative of that lineage. Polygala cruciata, the drumsticks, is a very common species across Eastern North America. It represents a northern extension of a clade that has whorled leaves. But we've got several species in that group here. Polygala rugelii, I mentioned this guy Randy years ago, the guy that found Polygaloides costifolia. This is Randy years ago. We were botanizing in Florida. Again, an EKU grad student. Polygala rugelii is endemic to Florida, but it's part of a clade that actually has it's a hotspot of like diversity for speciation within polygalas in the southeastern U.S. We have 23 polygalas in the southeastern U.S. So it's a center of diversity. And this series Decarinthes is endemic to the southeastern U.S. Polygala polygamy represents a separate clade that actually has got 3 species that are characterized by having what's called cleistogamous flowers. And by cleistogamous, we mean closed flowers, hidden flowers below the ground, under the soil. It puts out little flowers that don't actually open up itself. And then sometimes it puts out little hidden flowers in the upper leaf axis as well. But there's 3 species in North America that are part of this kind of unique lineage. And for me, I saved the best to last, the weirdest to last, Polygala seneca. One that's called snake root, lots of plants called snake root. It turns out that according to DNA, Polygala seneca is not related to anything else in North America. Polygala seneca is actually, believe it or not, any takers on where it comes from, it's Brazilian. How the hell did this Brazilian Polygala end up in North America? We don't know. But we've got an endemic species in North America whose closest relatives are all from Brazil. And so this is actually still an ongoing project, a project that we're still working on. There are people who thought that there should be at least 2 different species, maybe or at least varieties. But so far, our preliminary analyses are not showing support for 2 reciprocally monophyletic lineages. We're not finding support for 2 discrete lineages yet. But this is still an ongoing project. In fact, I'm going in June to a DNA workshop in Kansas with another EKU grad, James Beck. Some of y'all in the room might remember him. He published it on Short's goldenrod with Dr. Callie. And so another EKU graduate who's actually now going to be training me, kind of updating me with DNA work, basically. I'm a few years out of date, and he's going to be teaching this workshop. And so we're going to actually address this Polygala seneca thing again through that workshop. I'm looking forward to that. So as far as my original research goals went, awesome, right? This is perfect. I feel like this is absolutely wonderful here. Got way more done than expected, right? 6 genera, 53 species, at least a dozen lineages, published the Flora North America treatment. Yeah, I accomplished all of those goals with field herbarium and lab work, right? And in terms of biogeography, just to summarize here, we've got this one amphitropical disjunct. We've got a bunch of lineages that are kind of northern extensions from Latin America. We've got one that's actually invasive, and then we've got this one that actually has its closest relatives in North Africa and Europe. So again, this is something that's still we're in progress, still working on, but we have a much clearer understanding now of what the lineages of Polygala are. For me, once again, kind of wrapping up here, just the last couple of minutes here of wrapping up, pointing out that for me, research is actually only has meaning if it's shared, like if it's being involved in community outreach and in education. We need to have an educated public, basically. And that's what the research for me is about. It's about learning to share with others, right? I wanted to make it clear, though, that I'm not a one-trick pony, that I've actually worked with lots of groups, blue-eyed grasses, which are actually irises, miderworts, the Loganiaceae is a family related to the coffee family, right? I've worked with those. I've got all of these projects I should mention, by the way, involve students. These are student research projects that I've been involved in. I've had students working with leaf anatomy of the polygalaceae. Dicanthelium, an absolute nightmare of a genus with grasses. I'm actually on the Dicanthelium working group. The mallow family, the hibiscus family, the Asteraceae family, something we've been working with. Barcoding the flora of Florida, DNA barcoding is kind of a pet project of mine. I've actually got a DNA bank with almost 20,000 samples that I personally have collected. And so this is something that I'm really excited by. Barcoding is kind of becoming passe because molecular techniques are becoming more and more advanced. But the idea that you can actually identify a species and figure out where in the world it came from, it's still a pretty cool idea. People are just using other techniques now. But we were using this in this project here to actually identify that there was this new invasive Mycania in South Florida that was actually from the Caribbean. Okay. And so there's a couple other ideas just mentioning passing here. If you're familiar with iNaturalist, the website for naturalists here, there's a student I work with. He's at Murray over in Western Kentucky, and I worked with him last year at Hancock Biological Station. He was an ornithologist who was wanting to learn about plants because he realized that they were important to understand, just to know his birds better. But he's been out doing a lot of work. He just found a species in Kentucky, Monticello. And so this is a project with the students. I'm going to be working with this student this summer to go ahead and try to get this published. He wants to try to do some sort of a molecular-based study to figure out where the Kentucky plants came from. This is an inventive thing from the Western US. There's another iNaturalist person, a citizen scientist here who's down in Louisiana. This top map here is a heat map of my identification. So I actually do a lot of community outreach service through iNaturalist, putting names on plants for others. That's my passion, is naming plants. And there was this weird mystery plant, could not figure it out for the longest time. But this guy reached out to me and said, hey, I've got this weird thing. I said, go back and take better pictures. And he did. And we were able to figure out that it was this thing called Jabirosa integrifolia, which had been documented back in 1901, had been documented from Alabama, but never seen since. But now it's starting to pop up here and there in southern Louisiana. And so I'm actually going in May to work with this citizen scientist to actually document. Because one of the problems with iNaturalist, there's no vouchers. And as botanists, we need vouchers. We need vouchers, right? And so I think it's on us, it's incumbent on us to reach out to these citizen scientists and help them make their work of more value, right? So, and then for me, it's a very important thing here to remember, just keeping an open mind to new approaches. So one of my students right now in intro botany is a non-native English speaker who's a foreign exchange student, who's a business major, who should never have been in my introductory botany class if she'd been thinking about what it was going to mean for her. But what I loved about it, what I absolutely loved about it, was when I said make a key, she came back into the flow chart. My first response is, that's not a key. My second response is like, that's actually pretty awesome. That's pretty cool that you took what you know, what you're comfortable with, and you rolled with it. So I want to make it clear that for myself, I'm open to new ideas. Like I know Dr. Callie here, I heard 2 or 3 years ago, he's working with a fungal endophyte of this Brachyalgum erectum, this grass here. This is a really cool grass that has this venation that a friend of mine refers to as shattered glass venation. It's a pretty easy grass to recognize, especially once you know to look for that, right? And, I would love to work with anyone who's interested in botany and needs botanical help, right? And the last thing here is a shout out to the herbarium. I'm not going to talk much about it, but at the basis of all botanical research are herbarium collections. We absolutely need well-curated herbaria, and I hope to be involved in that. And then so finally, just a reminder that my primary passion is teaching plant ID. I like research, but research should be part of education, in my personal opinion. And this is just an example of a motif sheet. Students in my classes that are being taught patterns, they carry this sheet all semester. This is their cheat sheet. These are the answers to all the questions I'm going to ask them. And their job is to learn to see. I'm trying to train them to see plants. And they're going to be holding all the answers all semester as we practice the language of botany. So I very much look forward to the opportunity of embracing new possibilities here. And if there's any questions, I'd love to entertain them at this time.
Richard, what do you know about pollinators for polygalaceae?
Bees and flies are certainly involved, but the truth is there's the studies that have looked at that. There are some studies that have looked at pollinator visitors, like floral visitors. The problem is that floral visitor is not the same thing as a pollinator, right? And so ultimately, no one's ever. I've only seen one study in Europe that actually proved that they were pollinators, and that was with bees. But floral visitors, flies are very common. Lots of flying insects, true bugs will visit the flowers. But pollinators have only been documented that I've read about in bees.
Well, the different colors imply that there may be different fidelities with the pollinators.
Exactly. But no one has studied that in that level of detail.
This is kind of a more general question about how you work with students, especially in research. I'm just wondering, what do you think of as being kind of a successful collaboration with students for you and for them? What makes it, end up being valuable?
So for me, ultimately, it boils down to, does the student learn? That's my primary goal, is does the student learn? It's not the publication, it's not the end result. I mean, those things are nice. But does the student in the project walk away with their head held high thinking, yes, I learned something there, and I find that useful. So I just had a student present 2 weekends ago at the Arkansas Academy. It was her first poster. She was so nervous the whole time. She's super, but when I watched her face as she's standing, I'm thinking, oh my God, this is really happening. I'm standing at a scientific meeting in front of a poster. That was priceless for me. So for me, it's the student sense of accomplishment. That's what I value. In terms of setting it up to make it work well, it helps if you can have funding to pay them for their time, because most students have to work. At least where I'm at now, students can't afford to not work. And so if you've got funding to pay for that, that's awesome. But otherwise, it's just the opportunities are there. For me, the biggest problem is finding students who are actually willing to work with plants, because plants to them are, yeah. Finding students interested in botany has been my hardest challenges. I don't know if there's any more to the.
I guess I could ask another one. What about, like, how do you work with them to ensure that they learn? So funding, you said, but what other like process do you use when you're collaborating with students to?
So my model for how to successfully be a great mentor is sitting in the room over here. So I'm kind of embarrassed to answer the questions with Ralph Thompson sitting there, but being a good mentor, hands-on, after hours, the availability of actually spending time with the students, it's priceless, right? So making time in my life to make sure that I go out in the field with the students, that I sit down in the lab with the students. You don't just say, go do this and come back in a week. You sit down with them, you spend time with them, you take them out, you teach them, you spend time with them. So hands-on mentorship for me is the key to success.
Richard, was curious about your tropical collections. Were you able to ship everything back okay and get things deposited?
Yeah, so I've only got about 2,500 collections from South America, and yeah, I've got dupes of most of those. About 15% of my plants I never got dupes of. Yeah, but the positive, what I got was when I collected, I had to leave the first 3 collections in country, and the next 3 went to Mobot. So 6. By the time I got one, I had to collect 7. So I had to collect 7 duplicates for me to get one. But I've got roughly 2,000 tropical collections that are not mounted. I've been waiting until I settle, and I'm hoping if I end up here, this will be the last job I ever take. I'm hoping that this will be my home for the rest of my life, kind of thing. But I've got about 20,000 specimens actually that are unmounted that are going to come with me if I come here, including about 10% of my tropical collections. Yes.
Throughout your research, did you have any big, like surprising, like aha moments that something you found just didn't seem like it was real, like it was just like.
It wasn't even a polygalaceae thing. It said, I wish I could say, oh, it was this great moment with polygalaceae. It was actually with the pawpaw family, Annonaceae. I don't know if you know pawpaw, it's a similar trilobe. It's a very common widespread species in East North America. Everyone had always assumed that it was a northern extension of this tropical lineage, this neotropical lineage. But there was this hint that maybe there were some things in Asia that might be related, and I wanted that. And so I was working. I had just done a favor for Peter Raven, who was the director of the Missouri Botanical Garden at the time. I was still a graduate student at the University of Florida. I had just collected 23 species for a research friend of his in China. And Peter Raven said, "Can I do anything to help you? If I can help you out, thank you." And I said, "I would love to get some Discipulum or Anicocenthellum from Malaysia." And a week later, it was sitting on my desk, because that's how powerful the plant connected Peter Raven was, right? So all of a sudden, I got this sample sitting on my desk and analyzed it. And it turns out that our pawpaw is actually another Eastern Asian, East North American disjunct. And that was this cool, exciting moment of, if it went from being this kind of northern extension of a neotropical thing to another Eastern Asian. That was for the moment for me as a graduate student. I was like, yes, that was the moment of super excitement. But then somebody else published it before we had a chance to. Somebody else did the same thing. But that was my most, I think that was probably my single biggest aha moment as a graduate student researcher. So, yeah. And I thank you very much for being here.
I don't have any questions from Greenland. So. Let's call it.